Up in northern Sweden, Ellen Bushell is doing all she can in the fight against a disease that kills hundreds of thousands of people in the southern hemisphere.
Här har vi då ett mygglab där vi odlar myggor så vi kan studera parasitens spridning.
Malaria is a terrible disease that in particularly affects young children and pregnant women who are more susceptible to severe infection and disease.
Det märks att de är unga, för annars skulle tjejerna ha börjat bita. De är inte riktigt mogna.
Parasites enter the body through the saliva of the malaria mosquito and land in the liver. One single parasite can multiply in the liver and become 10,000, which are then released into the bloodstream. Here it is the spleen's job to constantly purify the blood. But to prevent the parasite from being cleansed out, it has a trick. It enters a blood cell, makes it sticky so it fastens itself in the blood vessels and can now multiply until it bursts the entire cell. When enough blood cells have been infected, symptoms appear: fever, chills, headache, muscle and joint pain, nausea. And without treatment, malaria can lead to death.
When I was 16 years old, I was really fortunate that I had the chance to go with my father to Tanzania. It really opened up my eyes to Africa, and that maybe I would like to do something with my career where I would have the chance to go back to Africa. But also if there's anything I could do with my career to make any sort of form of difference.
This interest in Africa remained, but transformed into working with diseases. Ellen began studying microbiology, and in a lab in the UK, something suddenly clicked.
The first time when I saw a malaria parasite under the microscope, it really was love at first sight for me. You can stain the parasite and color it in different ways, so you can really see the intricate structures and biology of the malaria parasite, and I knew that's what I wanted to study.
After almost 20 years in the United Kingdom, in 2018, Ellen chose to move back home to Umeå and the Institute for Molecular Infection Medicine.
What you can see here is EMAP3 in cyan blue, that it's very closely associated with the periphery of the red blood cell. And this is
There are drugs to treat malaria, but recently scientists have begun seeing that the parasite is developing resistance to the most effective medicine. This resistance is already widespread in Southeast Asia, and there are clear signs that it is also happening in Africa. So new solutions for eliminating malaria are urgently needed.
The malaria parasite has 5,000 genes, and we now know which of these genes are important for causing infection in the blood and also causing disease. What we are trying to do now is we're trying to understand exactly what these genes are doing. So what is the role they play in and what is the molecular function?
Ellen and the team knock out or turn off a number of different genes in different parasites. All genes produce proteins, some more important than others for the parasite to anchor itself and be able to multiply in the blood. The altered parasites with different genes turned off are put into the blood. Now it will be clear which ones cannot stick. Then we'll understand which gene sequence that was turned off was necessary for the cell to become sticky and get stuck in the bloodstream.
We really try to understand, what does this gene do? Where does the parasite get stuck when it grows? Where does the protein go in the cell? So we really try to study the gene in great detail, and that takes a lot of work. And this is also the goal with our research. We are trying to add one piece of the puzzle at a time to understand how the malaria parasite works. The work we do is basic research is curiosity driven. We are trying to understand how the malaria parasite works. However, the ultimate goal is that the discoveries we make can be used to develop new methods to fight malaria.